Welcome to our latest episode of the Lebanese Physicians Podcast. This is episode 110. And today we will be discussing 3D printing of organs for transplant. And our this episode's co-host with Dr. Muhammad Ali Jardal, and our guest is Kamil ElKhoury, who is currently based in Abu Dhabi at NYU Abu Dhabi, and has been recognized in the Forbes 30 Under 30 and the MIT Technology Review Innovator Under 35 Award. Camille, first, congratulations on this award, and can you tell us, I mean, how did you get into the field of organ engineering and bioprinting, and maybe give us also a background about yourself, how you got there, and the background about the field of organ transplantation.
Yeah, thank you so much. First of all, thank you for having me. It's really a pleasure. So, when I was in high school, I joined the Lebanese Red Cross. So, getting in direct contact with patients really gave me this joy of helping others. And once you get there, it's hard to go back. So. I was really good in all scientific fields, so I really didn't know what to choose for my major, but I was attracted by engineering. So that's why I chose to do chemical engineering at the University of Balamand. However, at the time, there was limited options to go. I was interested in going into the pharmaceutical industry to do research. There was limited access for that in Lebanon. So there was not really masters in chemical engineering and like sort of a minor in pharmaceutical or biomedical field. So mainly they were giving courses in petroleum engineering. That's why I decided, okay, it's time. I need to go do my master's abroad. So that's when I moved to the University of Birmingham in the UK to do my master's in chemical engineering. And there I specialized into pharmaceutical in the pharmaceutical industry. And there I found out about drug delivery, tissue engineering, 3D bioprinting, all the exciting stuff that I am doing now. And after that, there was an opening for a PhD position in France, which I applied and I got. And during my PhD, I did two international research mobilities, one at the University of Aveiro in Portugal, and the other at BWH and Harvard Medical School in the States. After that, I did one more postdoc in France before joining NYU Abu Dhabi for the second postdoc.
Amazing, amazing to hear. Can you tell us more about how you ended in organ engineering and bioprinting? I really want to know more about that. And if I'm honest, 18 years ago, I'm not that old, but 18 years ago, I dabbled into tissue engineering, but the field was not mature 18 years ago. And I didn't end up pursuing it much more than that. So I'm really interested to learn from you about your experience and how you ended up in that field.
Yeah, so it's still not very mature. There's a lot of advancements happening. So I took a course about tissue engineering during my master's, and my PhD was in tissue engineering. So when I went to France, we wanted to work on biomaterials for tissue engineering applications, and we wanted to combine nanoparticles with these biomaterials to create sort of a third generation of biomaterials. So the first generation was you put bio-inert biomaterials and you implant them in the body. They just give you support, but they don't give any bioactivity. The second generation, they will give you some sort of bioactivity or drug delivery. And now the third generation, we want to fully integrate the biomaterial with the tissue and to really enhance the regeneration of the tissue by itself. And when that happens, the biomaterial should really degrade and disappear from the body. So that's when I went to France, we started this idea of working on one type of biomaterials that I developed in Portugal and the States, and I'm actually still working on that. So a lot of development happening there. But in addition to biomaterial development, there's a lot of development happening in bioprinting, which you take this biomaterial, you put it in this machine, you give it a CAD design, and it will print it. It will give you the structure that you really need. And in tissue engineering or organ engineering, the structure is very crucial because it will affect the functionality at the end. And that's why we're also working a lot on 3D bioprinting now.
This is very exciting. It sounds like science fiction, something like straight from Star Trek. And like to hear you working on it and like have real world applications, it's something else.
So basically, Camille, to simplify it, so basically what you're saying is you get cells, correct me if I'm wrong, you get cells from the individual who you're going to do the transplant for, you use biomaterial, you combine, put these cells in the biomaterial, and the biomaterial lets the cells grow. It's like you're putting them in like soil fertilizer, letting them grow. And then now what you're doing, not only are you getting the organ, but you're also doing 3D printing to get the appropriate organ for the appropriate person.
Yes, exactly. So the idea may be that we will be able to do 30, 40 years from now. I don't know the timeline, but the idea is the patient will come to the hospital, right? They'll take a biopsy, isolate the required cells, proliferate them in the lab. At the same time, we'll do imaging for the injured area or organ. And from that image, we retract the CAD file or the CAD design, which is basically the design of the organ that we will give to the printer. So when the cells are proliferated enough, so we have enough cells, we combine them with this bioprinting, because this biomaterial, because the biomaterial needs to mimic the extracellular matrix that is usually present in our body, and it gives support to these cells. So our cells are not floating around in the body; they're fixed within this extracellular matrix, and it will give them these three-dimensional structure where they can really grow and proliferate more and have the similar function to what we found in our body. So that's the idea. We print the organ, which will be from the patient's own cells, and it will be it will fit exactly the injured area. So that's what we call patient-specific scaffolds or patient-specific tissues, because every injury is different. It will have a different structure. So that's the idea for the future. Right now, we're working a lot with stem cells. We're testing our biomaterials with stem cells, bone marrow or adipose derived stem cells. Also, there is a lot of development happening in cell engineering using iPSCs currently, which is really, really exciting.
So basically, just to give people an idea, and then I'll let Muhammad Ali go in. Basically, right now, for solid organ transplantation, you have to get an organ either from a diseased or passed away person or from a live person sometimes. But for the most part, it's a diseased person. You have to like fly over, bring it in ice, put it in the patient. It may not fit. It may not be exactly the same shape that you wanted. Then you have to put the patient on drugs that decrease the immune system so that the patient's cells do not fight the cells coming from outside. But what you're telling us is maybe if we are able to do this and you're using the patient's own cells, then maybe you don't even need the medication to suppress the immune system. And the next part of it is people with spinal cord injury. There's no treatment for them right now. So this could be a treatment for that.
Exactly, 100%. Because if you're deriving the cells from the patient, if they are the patient's own cells, there will be no rejection because now you're taking these immunosuppressant drugs, but like studies show that after 5, 10, or 15 years, there's a rejection happening for the transplanted organ. So in theory, there will be no rejection. The biomaterials that we use, we try to use natural sources that will not cause immune response from the body. And it will have now a lot of labs are working on immunomodulatory biomaterials so that they can also tweak the immune system into more proliferative response instead of inflammatory response when you implant those biomaterials in. But yeah, that's the main aim. of organ engineering is to replace the donation that is required now. But also for tissue engineering is very important because if you have a volumetric muscle loss, for example, where the muscles will not regenerate by themselves, you need to put something in place for this big area to regain function. Same for big bone fractures. If where will you get no one will donate bone. You need to get it from somewhere and to replace that. Same, for example, for sciatic nerves. If you have a sciatic nerve injury that is bigger than 4 centimeters, it will not regenerate by themselves. So even we can put biomaterials with bioactive molecules in that void, and it can help those tissues to regenerate in the body. So even without taking cells and proliferating them, the biomaterials by themselves, they can be bioactive.
Yeah, I mean, what you're talking about, if we're able to get there in the near future, it would be ideal. I mean, you have two solutions, either something like off the shelf, like a biomaterial that you just install, or you can customize the solution based on like the own body. So you have two pathways, and if you're able to do that, then you don't need organ donors, you don't need to go into all of that rejection. So really, it's the dream. And if we're able to do that, the applications are limitless. Even type 2 diabetes, for example, if you can like replace the pancreatic tissue, you won't even need insulin. So in terms of applications, they're really limitless. And it's really exciting to hear you talk about that. Can you share more about what your lab is doing? And can you share more about like the successes you're having and the recent developments?
Yeah, sure. So what we are trying to do really is to combine nanotechnology with these biomaterials to give extra function to the biomaterials. So, for example, I tried to merge liposomes with exosomes to create a new type of nanomaterial that can be used for targeting drug delivery applications when included in the biomaterial. I know I'll try to explain it simply. So the liposomes are the same technology that was used in the COVID vaccine. So you have the mRNA. You cannot really inject the mRNA by itself in the body because it will not reach the target. So you need to encapsulate it in these lipid nanoparticles and inject it in the body after that. So for that, I think the light went out. So for that, so there's a saying that it's not a drug until it's delivered, right? So if you cannot deliver the drug where it's supposed to be, it will not do its function. At the same time, our cells produce their own, let's say, lipid nanoparticles. They are called exosomes, and the cells use these exosomes or extracellular vesicles to communicate between each other. So liposomes are synthetic. They are FDA approved. They are widely used and understood since the 60s, and we can engineer them as we want. On the other hand, exosomes are newly found out or newly developed, but they are natural and they can really target specific tissues. So I tried to take advantage of the advantages of liposomes and the advantages of exosomes by combining both of these nanoparticles, and creating what I call the hybrid nanoparticle. And this hybrid nanoparticle I included with my biomaterial and tried to bioprint a complex organ. So what we try to do is to print a cardiac patch, so we can stick it on the heart, and it can release slowly the drug that we want. We were really targeting after a myocardial infarction. There's an injured area on the heart, and the cardiomyocytes, or the cardiac muscle cells that really beat and pump the blood throughout our body. These cells, they die after in that injured area, and they do not regenerate by themselves. So to cover this void, the other type of cells that is normally proliferating, which are the fibroblasts, or like it's their fibers, they come and they spread. So you have this area that's not really pumping, which is also problematic. And most of the people that survive the myocardial infarction, they have a lot of complications later on, and the doctor will tell them, don't do any sports or something heavy on your body. So that was one of the projects that we did. The other one was we really wanted to target. There's a big challenge now in tissue engineering and organ engineering is vascularization, because if you produce a big organ, you need to bring the nutrient to the cells in the middle of the organ, right? How does this happen in our body? Usually
through blood supply.
Exactly. So to create these blood vessels, it's very complicated, especially the small capillaries. Very complicated, and no one was able to do that. So you produce a big organ, the cells in the middle die, and then it's going to waste. So really, if you can produce these blood vessels from the big vessel to the smallest capillary, there you can solve that problem. And it's still a big challenge. So for that, we chose, let's work on something that doesn't require blood vessels in it, and that was cartilage. So we worked with targeting the meniscus tissue. And for that, we faced one problem, which is the mechanical properties of the meniscus that we printed. Because we work a lot with soft gels, because they are the one that really make the cells grow and move freely. However, the meniscus is a hard tissue. And so for that, we needed to work on the chemistry side of the material. And we instead of gelifying our gel one time, we double cross-link it with another solution to really match the mechanical properties of the human meniscus. And we did a lot of other studies, like to understand the immune response. When we put it in our body, we did antimicrobial tests, and it was really a great project that was recently published, and we were very happy about it. And that's where, using that, I got the MIT Technology Review Innovator Under 35 Award with the menisco gel, we called it. So yeah, hopefully that one we can see it on the market one day.
Inshallah.
Yeah. So basically, you told me like this was published in like in a journal that had an impact factor of 23,
Yeah.
which is very high. That means it's very highly cited journal.
Yes. It was challenging to publish there, but we made it happen. So we're very happy about that.
And just to explain it to people, so this menisco gel is like instead of, let's say, if you have a meniscal injury, right? You go in, you do the surgery, and maybe you replace the meniscus with muscle tissue or whatever. Basically, what you're saying is you don't have to do that. You just insert the gel in.
Yeah, so you can replace the whole meniscus. So what we did is we printed human-sized meniscus, and we encapsulated stem cells in the meniscus, and we were able to differentiate them into chondrocytes. So that really is, and if you can take these stem cells from the patient, or you take chondrocytes and proliferate them, and 3D print the meniscus. And what we basically do, we can take imaging for example, if the injury is in the right leg, we can take from the left knee the imaging and twist it and 3D print it. So it will be really patient specific, because a tall person will have maybe a bigger meniscus than someone short, or like everyone is different. So that's the cool thing about the printing technology.
Yeah, and basically you would be able to see, like, if somebody plays sports,
Yes.
will they go back to baseline? Because now a lot of people do the surgery and then they still have pains throughout their life, and maybe this will limit that if that happens.
Yeah, that's our hope. So now we did all the in vitro studies, and they were amazing. The mechanical properties match, morphological, structural, rheological, and biological properties. So now we're going to in vivo, and for meniscus, you cannot do it on rats or on mice because it doesn't exist there. So we need really large animals, which is a little bit challenging, but hopefully we get there.
Amazing. And you have a provisional patent on this technology, from what I understand.
No, not yet. So we're working with the NYU office to file
Okay.
that. Yeah, but that's very important. Usually we have 1 year for the US provisional patents. We have 1 year after the publication of the article. So it was published last month. So we still have 11 month to go.
Amazing, amazing. And the opportunities to commercialize that are limitless, really.
Yeah, hopefully that's our hope. So in the MENA region, now there's a big push for commercialization and for research related to that, and we hope we can achieve that for this project.
And basically the other, I mean, this is a great one. And the other one that I was interested in is what you were saying. If somebody has a heart attack, or typically what happens, you get scarring of the area of the heart where the attack happens, and then people get heart failure. So basically, what you're
Yes.
saying is you can inject a patch or put a patch in using, uh, correct me for nanoparticles,
Yeah.
liposomes and exosomes, and
Yes.
and you are able to prevent the scarring. Basically, you're putting something to prevent the scarring from happening, so the heart remains functional. Is that what you're saying?
Yes, exactly. So now there is in vitro, there's a lot of cocktails that are being used to where they call them reprogram the cardiac fibroblasts back into cardiomyocytes.
Wow.
But that happens in a dish. When you inject those chemicals in the heart, it's not working. There's two reasons for that. First, it's not being delivered inside the cells. And second, you need a sustained long delivery. You don't need to put it, so either you need injections every day, or you need a technology that will release this chemical or microRNA cocktail over a long period of time. And that's what we are trying to achieve. So we embed these cocktails in the nanoparticle to assure the delivery inside the cells. And we embed these nanoparticles inside the gel that we stick on that injured area to make sure that it's going to be released over a long period of time exactly at that area. Because if you inject it in the blood, it will go everywhere, right? We need it on the injured area. Liposomes, we use them because they are well understood, FDA approved, and you can encapsulate big amounts of drugs in them. We combine them with exosomes because exosomes can really target cells and tissues. So if I want to, in the heart, there's many types of cells. You have endothelial cells, you have cardiomyocytes, you have cardiac fibroblasts, you have macrophages. But I want my cocktail to go to cardiac fibroblasts and transform them into cardiomyocytes. That's why I take the exosomes from the cardiac fibroblasts, and then they have these proteins on their surface related. It's like a key with a lock, right? If you take the exosomes from one type of cells, it will only enter this type of cell. And that's what you are trying to do. So sustain the release over a long period of time and really target the cells that we want.
So usually your innovation is twofold. The first step is in terms of the drug delivery, and the second step is the drug itself. It's not a drug per se. It's really this cocktail of nanoparticles, if I understood you correctly.
Yes, so this is not our discovery or innovation. It has been done since 2012.
Oh,
Yeah, so but it was only done in a dish and not really because and the efficiency is very low. So that's what we are trying to work on. To work on the technology. Like I said, if you do not deliver it, it's not a drug. I mean, it can do anything it wants in a dish, but if it doesn't reach where it's supposed to reach in the human body, it will have no effect.
okay, okay, fair enough.
Yeah, and I imagine it has to be done like immediately after the heart attack, because if you wait, then the tissue dies, and it's going to be very hard to regenerate at that point in time. Because once
Yeah,
the
and the
scarring
and
sets in, you can't
yeah.
reverse it anymore.
Yeah, and the fibrotic scar gets denser and denser
Yeah,
because these cardiac fibroblasts they proliferate very quickly,
yeah.
and it will be harder to reprogram.
Mm.
And are you working on animal models at this stage, or?
So here we have rats and mice that we are trying to do. But yeah, this project we are continuing now with because I did it at BWH and Harvard Medical School, and we're continuing that over there.
Amazing, amazing.
It's still going.
And so how were you selected for the Forbes 30 under 30, and how were you contacted in regards to that?
So there was a call for application, and the university really encouraged all of us to apply. And they said there will be a committee that will really study case by case, and they decide who to award it to. And I was very lucky, along with two other people from here, to be selected for this year.
And
the MIT Technology Review, that you also won that recently, correct?
Yes, correct. So the MIT, actually, it was a friend who encouraged me to apply. He got it last year, and he told me, no, with your profile, you should definitely apply for it. And yeah, I did, and I was also very happy to get it.
Mabruk.
Thank you, thank you. Yeah,
so
these
Kamil,
awards, I mean, they have they increase the visibility of our research for sure. A lot of people contacted me to understand more what we are doing. Some of them proposed to collaborate, and we're trying to work it out. But also I felt it with the students at the university. They also are more interested. Can we join your lab to do some research on that? And it also kind of validated our work. It's like encouraged us, like you're going in the right path, continue. So it was really good.
and Kamil, what are your next steps? Because I mean, I think this field is limitless. I mean, there's a lot of things that can be done. What are your next steps in the process?
So I really want to focus my lab into three key areas. First one is a combination of nanoparticles with biomaterials to create new combinations, because we can give a unlimited amount of properties to these biomaterials. One property that we didn't mention is the electrical conductivity, which is very important for heart tissues and neural tissues, for example. So that's one area. The second area is the biofabrication techniques. So I talked about bioprinting, but that's not the only biofabrication technique that exists. There's a electrospinning, and now there is in situ bioprinting, where you do the bioprinting directly on the patient. We're using a robotic arm. It's something really from the future, and you scan with the phone. You scan the area with the phone, and then you transfer it by Bluetooth to the machine, and the robot will make the printing directly on the patient. So really a lot of development happening there. So I also want to work on new biofabrication techniques, or combine techniques that already exist. And the third one is to work on not just organ engineering or tissue engineering for implantation, but also for drug discovery. So what happens now is that a lot of drugs fail in the last stage, and we can really tackle that by testing them on real human tissues produced in the lab. We can really predict the efficiency that they will have later on, instead of going into small animals, large animals, preclinical, clinical trials, phase 1, phase 2, phase 3. If we use really the same tissue in a healthy sample and a diseased sample, so create a healthy cardiac tissue or a diseased cardiac tissue and test the drugs and really check the efficiency, we can save a lot of time and a lot of money. So these are the three key areas I want to focus on. So they are each their own areas, but they also can be combined, which is very exciting.
And how is it being in NYU, Abu Dhabi, and you're still collaborating, from what you told us, with the lab in Cambridge? How's that, like, cross-continental collaboration?
So that's NYU really is the best environment that you can find for research. So we have state-of-the-art facilities, and really they really acquire the latest equipments. So if there is any upgrades, they don't say, "Oh, we got it last year." No, they get the upgrades. There's a huge investment into science and technology now in the UAE, which is also very exciting. And there is a push for commercialization, which really motivates us, because at the end of the day, if you only do research to stay in the lab, you'll lose motivation. So that's not what I signed up for. I signed up to help people, and I really want to see my work helping people at the end. Otherwise, there's no use in doing it.
Kamil, it was great talking to you, and I want to thank also Muhammad Ali for connecting all of us together. I think this was a great episode, and I think I
Exactly.
learned a lot from this. This is a field I don't know a lot about, and I learned a lot, and it's a pretty exciting field, I think, in the future. So we're glad to know you, and I hope we do this podcast again, and then you'll be like a very well-known person around the world at that time.
نوبل خلص نوبل برايز ما حنقبل اقل من هيك.
Yeah, thank you so much for having me, and I'm always up for it. We can in later episodes we can discuss really projects in details, and I try to explain them in a more comprehensible way. And thank you so much. That was really great.
Thank you.
Thank you. يعطيك العافية.